Today, we're going to talk about the phototransduction in the rod cells of the retina. Inside the retina, there are two different types of cells. There are cone cells, which see color, but we won't talk much about them today. And then there are rod cells, which see light. Under a microscope, rod cells look a little like this. When you zoom in to the rod cell, you're going to see a few different things, but most importantly for right now, you're going to notice these blue discs represented by the blue sour strings. Now the discs themselves aren't blue, it's just what color candy I had. On the discs inside the rod cell are these molecules called rhodopsin. When you zoom in on rhodopsin, you're going to notice a few different things. First of all, you're going to see that rhodopsin is broken down into 7 different compartments. Now, the protein portion of rhodopsin is called opsin. Opsin makes up a little pocket which houses 11-cis retinol. When a photon hits a rod cell, it's going to come in and also hit rhodopsin, which in turn is going to make the 11-cis retinol change shape. When the retinol changes shape, it's going to make the rhodopsin change shape as well. When the rhodopsin gets too big for its britches, it's going to make this G protein, also known as transducin, pop off. Now, transducin, when it's inactive, is bound to GDP. But when it becomes activated, it gets to exchange that GDP for GTP. When the G protein pops off of the rhodopsin, one of its 3 subunits, its alpha subunit to be specific, is going to break away from its 2 brothers and attach to phosphodiesterase, which is this yellow candy right here. Now, the phosphodiesterase is responsible for taking cGMP and transforming it into just GMP. When the rod cell is depolarized, the cGMP is responsible for keeping these sodium ion channels open continuously. These channels allow calcium and sodium ions to flow into the rod cell. However, when phosphodiesterase takes the cGMP and transforms it into GMP, then the cGMP concentration levels are lowered, which in turn causes these channels to close. When these channels are closed, it restricts the flow of sodium and calcium ions into the cell, but does nothing to keep these potassium ions from flowing out. When these potassium ions flow out, it is going to cause the cell to become hyperpolarized, which in turn sends a signal to a bipolar cell, which will then send another signal, which sends a nerve impulse to the brain, allowing us to see. Now, after all this happens, the cell kind of has to reset itself. And it does this with the help of guanylyl cyclase. That takes GTP and transforms it to GMP to replenish the levels within the rod cells so that these little channels can be reopened, allowing the sodium and calcium to get inside the rod cell, which will cause the cell to become depolarized once again. When it's dark, the rod cell is depolarized, but when it's light, the rod cell becomes hyperpolarized, allowing us to see. Thank you.